I will continue with the agenda. As you can see in the agenda, we have many interesting presentations from companies from Austria, Romania, Germany, and Spain today. The agenda was sent out in advance, and each participant will be presented before each presentation. And now I would like to hand over to my colleague Gavel Bartek, who will now present an insight into the DIGI-B-CUBE project and the associated funding opportunities for you. So Gavel, the stage is yours.
Thank you, Patricia. Good morning, everybody. I'm really glad to be here with you today. And my role today is to just shortly present you our project and mainly opportunities which are related with our project for you. I will share my screen and. Yeah, so let me check now. It should work. Yes. So DIGI-B-CUBE. DIGI-B-CUBE is the reason why we meet today, and the project is funded by European Union from Horizon 2020 program, and it aims to foster digital innovations in the medical diagnostics value chain and related value chains through the promotion of cross-industry cooperation between companies from healthcare and IT sectors. We are 9 partners from 7 countries, and these countries are Austria, Belgium, Norway, Spain, Greece, and Germany, which you may find interesting, this information also while thinking about potential partners for you. So what we can offer in our project, what we have is digital maturity analysis for companies, matchmaking with potential partners from both sectors, innovation projects. We have funding to grant innovation projects, and coaching after those projects are delivered. So these are the two sectors in how we see them. So for healthcare is biobanking, bioimaging, biosensing, and others. And for IT is IT like artificial intelligence, big data, apps, IoT, but also other technologies related to electronics and robotics, or focused on digital innovation. So here the main message is that both definitions of health and IT are really broad and general to include as many technologies as possible to make those innovation projects more inclusive and more innovating. This is how we see those two sectors. This is also how we see the potential interconnections between them, which you can also may find useful while thinking about your ideas for potential projects. Regarding the granting scheme, we have different types of vouchers with maximum amount of grant per SME with 60,000 euros. And here the most important thing are the eligibility criteria, which is compliance with the EU definition of the SME status, location in the European Union country or Horizon 2020 associated country, and to be active in those two sectors which I mentioned, IT or healthcare. These are the vouchers, these are grants which we have for types, one for travel to attend events which we organize. Then the second one is for prototyping with maximum value per voucher 20,000 euro and maximum funding per SMEs to 20,000 euro, 60,000 for per project. Then another type of voucher is customized solution innovation, up to 50,000 per SME and 150,000 per project. Both include cross-sectoral consortia with minimum 2 partners from health and IT sector. Prototyping projects runtime is between 1 and 3 months, and for customized solutions is up to 6 months. The eligible costs for those projects are personnel costs, subcontracting, and VAT according to the national legislation. This is the procedures and the project lifetime. So now we are with the first open call published in April, and with deadline by the end of July. So of course we invite you to apply with your ideas now for this call, and the results will be published in August, and projects will start in October. Then we have second round with second deadline in February next year, and again the evaluation procedure and the selection of projects by March, starting of projects few months later. So this is all from my side for now. Here's some information about our next events, but the main message, of course, for you is that we have this granting program, we have funds for you, we have also our networks and contacts with companies with both sectors, which may be your potential partners. So please do not hesitate to contact us, and enjoy the webinar today. Thank you.
So thank you, Gavel, for the presentation, of course, and all questions about the call, please go to the chat, where there also be the answers found. So our next speaker and first representative of an IT company, it is Florin Kardasim from the company Strong Bites Consulting, and he will give us an insight into the diagnostics of heart diseases by means of machine learning. So Florin, we look forward to your presentation, and the floor is yours. Florin, you have to unmute yourself if it's possible.
So hello everyone, I'm Florian, and I'm sending you my warm regards from the beautiful city of Iași. I'm the co-founder of an IT company which is called StrongBytes, and first of all, I would like to uh, congratulate the entire DIGI-B-CUBE team and the partners for the overall management of this project, especially given the current crisis context, actually. So guys, you're doing an amazing job. And thank you so much, of course, for having me today and for the opportunity to get connected with like-minded people all over Europe. My short intervention today will be about heart diseases diagnostics using machine learning, which is actually the project we're about to submit to the Customized Solution Innovation DIGI-B-CUBE call. So we've got a lot of love here in the city of Iași, and it's not just because we're kind people, but because the medical science and technology make a great deal together through the collaboration of the medical and IT universities, clusters, and companies. Well, an old and wise AI agent once said that should heart love the brain, then there is a big chance that the brain may love it back. So this was a source of inspiration from this AI agent for us. So what we've done, actually, we've grouped together with some other two companies planning to build a next generation product. So we've seen the AI increasingly being involved in the medical act with lots of advances in machine learning and novel capabilities for predictions and diagnostics in the cardiomedical field. So the big players such as Google, Microsoft, Amazon, renowned universities, research centers in the world, and yourselves, of course, continue to amaze us with outstanding results. But obviously, there's still a lot to do in this space. So as part of the Imago Mall Regional Innovation Cluster in Iași, we formed a consortium consisting of one of the top medical companies in Romania in medical imaging field. This is ConExpert, and two IT companies specialized in cloud, data science, and machine learning. And this is Romsoft and StrongBytes. The two main challenges that we're tackling with this project are the facts that data is still siloed and difficult to access in due time by the specialists. And the second one is that good diagnostics take time and knowledge. And here is where a solid digital infrastructure and machine learning can help. Our goal is to advance and implement our existing product by providing easy-to-use, accessible DICOM web interfaces and to help doctors identify anomalies and make diagnostics. Of course, it's a huge space to be explored, but for now, we're narrowing down the focus on three specific directions. Number one is coronary arterial dominance. Number two is coronary origin anomalies. And then last but not least, number three is coronary calcification. So this is what we're going to focus on in this project that we're applying with in the DIGI-B-CUBE call. That being said, thank you very much for having me, and good luck to everyone who's going to participate in the call. Also, we're open for business, so please don't hesitate to drop us a line if interested in the results of our research or having any other particular, I don't know, opportunity to discuss. So thank you so much again. Have a great day, and let's enjoy the rest of the day.
Thanks, Florian, for your great presentation and also to the congrats you gave to us and the DIGI-B-CUBE project consortia. So as before, all questions for Florian, please go to the chat. They will be answered there as well. So now I would like to move on to our next speaker, Max Sonnleitner from the company GENSPEED. He will tell us today about the testing of infected and healed COVID-19 patients with his developed test system. So Max, the webinar stage is yours. Thank you.
Yeah, thanks, Patricia, for inviting me and for hosting this. So it's my pleasure to present to you our platform, and so it's a rapid point of care testing platform. And especially I want to present today what we are right now developing, which is a rapid test for detection of in the topic of COVID-19. So there will be 3 tests: antibody, antigen, and a combined test. And I want to run you through those things. So with regards to the company, so we are a startup company. We spinned out our company in the end of 2016 from Greiner Bio1, which you may know. It's a big company doing blood sampling in Upper Austria and worldwide. And we are headquartered in Rheinbach, a small city in Austria. At the moment, we are 10 employees. And we are focusing on development, production, distribution of rapid tests for various applications. And the basis is the GENSPEED technology platform, which we developed under the umbrella of Greiner Bio1 starting in 2009. So we have 3 key product areas, one of which is using the GENSPEED technology for next generation point of care testing. So anyone that has a lab test that he wants to transport to the point of care, then we have a perfect platform for doing that. We have everything is IVD-CE certified, so it's certified in Europe as a medical product. We have 2 products on the market right now. One is chairside test for fast detection of key pathogens in periodontitis, and the other one is a test portfolio in hospital-acquired infections. But today I want to talk about the COVID-19 testing that we plan to have also as a portfolio. So our platform technology is based on 3 components. First is a microfluidic test chip. Then we have a small device where you put in the test chip, and then we developed also software. So and now we are really developing 3 new tests for COVID-19 testing, and maybe it's interesting to see where we want to do the testing with regards to the onset of the symptoms. So here you can see the lines that represent more or less the DNA or RNA material in this case for the virus, that the concentration increases when the symptoms are set on. And after 10 days, 10 to 14 days, you get the seroconversion, and then you get the antibodies. So that's those lines here. That's the green one is the IgG. And in the beginning, as you know, you can do the PCR-based tests that detect the DNA, and then after 14 days, you can do the antibody detection. So that's the diagnostic window for this, and what is out there. So there are the PCR-based lab tests that are really time-consuming, 3 hours usually, and then you have got the antibody tests. There are now lab tests available, and there's also the strip tests. The lab tests have good quality, but the strip tests have questionable quality. And this is where we wanna hit the market with a new, really sensitive, highly sensitive and specific antibody test. And then we will have also an antigen test. And the final product will be a combined test. That means that you can really test for virus infection and antibodies at the same time on one test tube. So that's the basis. We are using the GENSPEED technology platform for doing that. As you can see, we provide a tablet with it. You can see this running with a countdown here. So in this case, it takes 12 minutes. This on the right-hand side, that's the test kit box that provides everything that you need for testing of the patients. So this is a short animation. I hope it works. So maybe if there is no sound, Also okay. So this is how the procedure is working. You take a finger prick, and you transfer this 10 microliters of blood into a small vial. Then we press the blood through a filter tip onto the chip. As you can see, it's then a transparent fluid, so it's blood serum separation that we do there. And then the countdown starts. In this case, 13 minutes. And then in the end, you get your results. So that would be COVID-19 antibody testing or also antigen testing. So just quickly, I want to show you the principle of the tests that we are using. So that the very most important thing is that we are not doing the lateral flow test strip technology that you know probably from pregnancy tests. We are really using an ELISA test procedure. That means we are really transporting the ELISA test from the lab to the point of care. So usually we print the viral antigens on our test chips, and then if you've got antibodies in your blood sample, then they will bind, and then we use a second detection antibody that gives light within our device, and we can detect. The very important thing about our device and our test now will be that it's compared to a lab-based ELISA test or to a strip test, that we use 3 different proteins for testing. So we are printing the spike protein, the N protein, and also other proteins from other coronaviruses, and then we are detecting the signals. So that means we can really see, is it really the new coronavirus, or is it another coronavirus where you've got produced antibodies? And the control shows you then if everything was correct. So the USPs, the advantages of the system are, of course, that you only need 10 microliters. You get the results within 15 minutes. You get 100% agreement with the lab ELISA test system. And we can also give you really high specificity as we test for different viral antigens. The test result will not only give you yes/no answers, but it will give you low, medium, or high levels of antibodies. And also, in addition, you will get information if your antibodies are really, let's say, protecting you from the virus, because we will see if they are against the spike protein or others on the virus. So that's the antibody-based test. As you can see here, for our first results for 25 patients, so we have 100% sensitivity and specificity compared to a lab ELISA. That's the first test that we will launch on the market in the end of July. And then we will have 2 other tests, one of which is the antigen test that tests if you've got an acute infection by testing for viral antigens, not for the RNA, but for viral proteins. So in this case, we will print onto the test chip the antibodies, specific antibodies, and if you have an acute infection, we take a swab material, put it on our test chip. If there is a viral protein there, then it will bind to the antibody, and then we will again have a detection antibody and generate light in this position of the chip and detect the light with chemiluminescence reaction, and then see if there is a viral antigen, if you have got an acute infection or not. What's very important to emphasize here is that we will not only test for coronaviruses, but also for other viruses. So we will put here the other coronaviruses as well, but also put here influenza virus, for example, so you really can discriminate what kind of infection is it. So concluded, you will have a measurement from swab material. You will have your result in 15 minutes with high sensitivity, because it's microfluidic and chemiluminescence based. You will get high specificity, and you will also get information about the amount of the virus particles that are in the material. And last but not least, we will have a complete solution where we put both tests together and combine them on one test chip. So this will be on the market next year. There's a patent still pending because we will change the microfluidics here, but it will be the first rapid test that will target both the infected and recovered patients by testing for acute infections and also for antibodies. And that's what I wanted to present to you today. And yeah, I mean, we are looking forward to collaborating within the DIGI-B-CUBE with others, with IT companies. And yeah, happy to answer questions or to get in contact with some IT companies. Thank you.
Thank you, Max, for the presentation and also for the perfect illustration with the video. So now we know a bit more about your developed test method and especially specialized on the COVID-19 virus. And also, or as already mentioned, if there are any questions for Max, so please go to the chat. And I want to introduce our next speaker. It's Alfred Wegerer. He has founded a medtech startup which is completely focused on lung function, and he will tell us more about this in his presentation with the title Artificial Intelligence for Better Respiration. So the webinar stage is yours, Alfred. And please unmute your microphone.
Oh, I can. Sorry. Thanks, Patricia, for the reminder and the nice introduction. Very welcome from my side here from Lake St. Wolfgang in the nice Salzkammergut in Austria. I would like to give you some insights of lung diagnostics, an early stage enterprise. As the name tells you, we focus on healthy respiration using artificial intelligence combined with a smart sensor fusion system. Florine gave us some ideas on AI could help in the healthcare industry. So I just want to give you a short overview. There's no total protection for our respiration system. It was an issue in the last 2 weeks, as everybody knows from you. I would say masks are not an option, even at that time. So we have to think about how to protect lungs and make diagnosis in a very early stage. You see here some bullet points like smoking is not the only thing which damages lungs, as we sometimes hear when we talk about COPD or other things. So there are many other things in our civilization like air pollution, aerosols, bacteria, or even viruses, as we got from the corona crisis now. An early detection of all these things would help to give a more positive prognosis and to avoid damages. But this would mean we need a solution which we can use in a very early stage at the GP practice and in point of care facilities. The few things available on the market are mainly very difficult to use and need well-trained staff and a high patient compliance to get the right results out. So we see our challenge in the main market of COPD and bronchial asthma at the moment, which is a €1 billion market today. And so we are not the high sophisticated solution for the clinics, which we see a solution which works at almost every home, works in the point of care, and is a very easy to use lung and respiratory tract monitor, like you know it maybe from the blood pressure monitoring system, which started in the 80s and is now available almost at every home. When we talk about AI, we see a very comprehensive solution when you talk about data analysis and machine learning, as we learned from Florine, as a basic environment to take out all the measures and the data and to give us the right diagnosis. Here's a little view on our device. So we do not talk about software only, but AI is a big thing also in our LUDI system. It's just a working title at the moment. In our environment, we have a device, we have a sensor fusion system, means a smart array of intelligent sensors, which we combine with the data fusion software. This is already an AI-based solution to calculate all the data and combine and compare it with existing information and to give feedback to the patient and to the doctor. But we use it on a very discrete interface, so the device has all the AI software on it, and the smartphone or a cloud database is just to work with the patient and the doctor together. Here on a first glance, the structure of the device with all the modules and a little picture of our first clinical trial at the Kepler University clinics here on the right side. Where are we now at the moment? So we have patent pending for the device, especially for the sensor array and the data fusion software. We did already the first clinical trial with 100 patients, and in this trial we got confirmed the sensitivity and the specificity of the device, and are now very active in building the serial product and programming the AI environment. Because this meeting is also to find partners out there on the market. Yes, we are looking for software programmers in the artificial environment sector, especially based on Python software tools. If there is somebody out there, would be happy to discuss this. As a coincidence, we have a technology partner producing for us the equipment based in Bucharest today, based on all the medical device standards. And a third very important part for us is always the clinical trial procedure. So we are in a very lucky situation in Upper Austria together with the upcoming Kepler University clinics. But we're always interested in spreading the trials and talking to other organizations as well. In the last weeks, we had a couple of requests and discussions with potential partners, but we see always an issue in the requirements in the typical medical device regulation standards. We want to go with the MDR, despite it's postponed to May 2021, but we think we should go with it all even today. And so we would ask to think about all the requirements in the medical device industry. Here's just a little list to get in mind what is necessary to go out with a product and to have a right partnering available. As I said before, we are an early stage company, but we did many scientific work the last 2 to 3 years, and are now very close to the first product version, which we want to release by the end of the year, and start the next medical or clinical trial for version 1. But we have in mind 2 next versions, version 2 and version 3, and see the next 4 to 5 years on our roadmap, which is mainly financed by public money and venture capital at the moment, but needs partners to get done all these things. So thank you very much for these few minutes. Let's keep our respiration system healthy is a slogan we have here with us. And I would be happy to discuss the one or the other cooperation out of our team here.
Thank you, Alfred, for the very interesting presentation, and also greetings back to the wonderful Wolfgang See in Austria. All questions for Alfred Wegerer, please go back to the chat, and there will be also the answers found. So now I would like to welcome our next speaker is Sebastian Alberg from the company Magnosco. In his presentation, he will tell us more about the company's own platform for epidermal data. And Sebastian, we look forward to your presentation, and the stage is yours. Thanks.
Thank you very much. Thank you for having me and giving me the opportunity and the chance to talk about Magnosco today. I hope you can hear me clearly. Just a few words to me. My name is Sebastian Albeck. I'm the managing director at Magnosco, where I belong, I would say. Since I'm a student, I worked on the detection of skin cancer and developed a device for diagnostic and treatment of skin cancer. And after that, I worked on the digitalization in healthcare, in radiology. And so I would say a good match for me, at least. So now you know me a little bit. Let's come to the interesting part. Let's come to Magnosco. Magnosco is a Berlin-based medtech startup, and it was founded in 2014 with the aim to improve the diagnosis of melanoma by means of laser spectroscopy and artificial intelligence. And we had a lot of achievements since the founding, such as the development and CE certification of our device called DermaFC. We had several financing rounds, the first go-to-market and the soft launch, and now we are working on the next generation of our device with focus on the US market. But why is melanoma of interest? I guess you all know that melanoma is the most deadliest skin cancer type, and its incident rate is increasing annually with about 300,000 new cases each year and 60,000 deaths. But early detection is key here and can save life. And we are early and earlier than competitors using image recognition or other technologies.
Sebastian, please unmute your microphone again. We can't hear you.
Oh, sorry.
It's fine. So please start again with this slide because then we lost you. Thank you.
I see, no problem. So then I start again with the overall problem and the reason why we found Magnosco. And this is that the diagnosis is very subjective and dependent on the experience of the clinician and is often of poor quality. And if you know the process of skin cancer screening, you may know the trouble you could have. You make an appointment with a dermatologist or in some countries with your GP. And if there is a suspicious lesion, a biopsy will be performed, which is a little surgery and often not on the same day. And the skin tissue will be removed and then sent to the laboratory. And it takes up to weeks to get a result from the laboratory. So weeks of stress for the patient, think about, may I have skin cancer or not? And in the end, 97 to 99% are false alerts. So although melanoma is a highly mortal disease, if it is detected early, the chance to cure it is about 95%. And so Magnosco's idea was to develop and approve a medical device. So we did that. We got the CE certification in 2017. And yeah, we had a hardware heavy device. And to be honest with you, it will not be commercially successful as it was never designed to be. But it brought us where we are right now. And I think that is very impressive. So Magnosco's solution is based on a non-invasive method. So no harm for the patients, no cutting. We have the highest accuracy on the market, and we create an objective score giving diagnostic support to the clinician within minutes. And there are big advantages for the dermatologist as well as for the patients. And now let's have a closer look to the technology. It's based on a method called dermatofluoroscopy, which is a specific type of laser spectroscopy, 2-photon excitation to be precise, using the melanin fluorescence. Melanin is a brownish pigment which is accumulating in the skin if you are in the sun. And we are the only ones worldwide who are able to excise and detect melanin. And the good thing is that there are tiny differences in the spectra if the lesion is malignant or benign. And what we are doing is scanning not just one point on the lesion, we scan the lesion pixel by pixel every 200 micrometers. And in the end, we get up to 1000 spectra per lesion with about 700 data points per pixel. So a lot of information which need to be evaluated. And for proper analysis, we use AI. And we do not have a live learning AI. Our AI was trained on around about 50,000 spectra during a clinical trial. What is the difference to image recognition as example? We just have one lesion there, but one lesion and one picture there. We have also one lesion, but we get hundreds of spectra. So a lot of information, a lot of data. And the data was collected at the well-known university hospitals of Charité Berlin, Heidelberg, and Tübingen. And let me shortly explain the study design to you. The suspicious lesion were selected by the dermatologists. It was scanned afterwards by means of our DermaFC. And the registration of the lesion as malignant or benign was due to the diagnosis of the gold standard, the histopathologist. And of course, the data were divided in a training set to design the AI and a test set for validation. And let's look a little bit more into detail. The lesions have not been crystal clear, as many others have done in their studies, and no dermatologist would need help by technology to try to diagnose them, as they are so clear. We took in-between cases, which need to be removed, as the dermatologist diagnosed them as possible malignant. And those biopsies have then been diagnosed by histopathologists. As I mentioned, they are the gold standard. But due to a lot of publications and scientific consent, histopathology is not as good as needed, can differ from physician to physician, so is objective, and therefore is questionable as gold standard. And that leads to a ground truth problem in the end for our data. And to increase the ground truth, we tagged our lesions with good or bad by the results of 3 independent histologists as a majority vote, so to say. So if you know that problem and are interested in discussing that, please feel free to contact us, as we have also other algorithm-based ways to address that ground truth problem. So back to the process of AI design. We pre-processed the data, the spectra, and then trained the AI by supervised learning using support vector machine classification to create a model. And this model was then validated on our test set and implemented on our machines with an outstanding accuracy. And of course, it is not the end. We talk about big data, and we're working on new studies to improve and increase our data set to design even better AI. And we have acceptance of the method by peer-reviewed publications such as Voucher et al. In the British Journal of Dermatology. We also won the scientific award of one of the biggest clinical network for dermatology in Germany. We showed the proof of concept during our soft launch in 2018. And Heidelberg published an article showing patients' trust on our method. By the way, the highest trust and what is interesting is that they showed that the highest trust was shown in the co-work of the clinician and our technology. So what we always claim, we do not want to substitute the expert, we want to support them. And we also have the potential in other diseases, interestingly, such as Parkinson, as neural cells can also have melanin in it. And the first investigation on that was published last year. And yeah, our science team is keeping on working on new studies and ideas the whole time. And I talked about the soft launch in 2018. We placed a few DermaFC on the market to have real-life testing of the devices, get QL feedback, and see once again the usability, and had promising results by that. But yeah, here we are having an incredible melanoma detection. But the question has to be asked, and of course, this is not just a scientific, but also a business question. Is there more possible? And the answer is totally yes. Melanoma was just the beginning and the entrance in the healthcare and medtech world, but it was really just the first, but of course, huge step. We took our time and learnings and ideas and input from partners and externals and reviewed the data. And yeah, we showed potential to increase the number of applications, as example in a publication from the Charité, which was showing that you can use our device for the diagnosis of pigmented basal cell carcinoma as well. And we have big data collection with a lot of molecules but melanin. By means of powerful AIs, we will solve other dermatological questions such as psoriasis, acne, or unpigmented skin cancer types. And the only thing what is missing is the commercially successful device. And that is what we are working at on the moment. So Magnosco is developing a technology platform for skin-based applications and creating a multi-application device for R&D, diagnostics, and prognostics. So Magnosco is a highly potent venture showing a cutting-edge combination of engineering and powerful AI, a highly motivated international team, and the support of great experienced partners as well. And I think most important, a huge market potential in the end. And by showing you our team and the heads behind Magnosco, I want to finish and sum up what we are doing currently. So as I said, our strategy is to enter the US market as the #1 market for dermatology worldwide, but not just the US, of course, but it's the #1 priority. And therefore, we create a new generation device, which is smaller and faster and cheaper in production and user-centric. And we're not on our own, but work together with experienced partner for hardware and optic development, and also collaborating with a strategic partner and performing market research in the US to match the requirements to be successful with our next level device. And we use our old devices in parallel for clinical studies to increase the number of applications and show the great potential of our technology, not just in melanoma, but other fields in dermatology or even cosmetics as example. So I guess Magnosco will become the first technology to really improve skin cancer and skin disease diagnosis and improve skin condition detection, but also become commercially successful. And yeah, with that, I thank you for your time and I'm open for questions in the chat.
Thank you, Sebastian, also for this very interesting presentation. And as already mentioned, all questions will go to the chat. Of course, also the interesting comparison can go to the chat. And I see that there is already one. Okay, and so far we have heard a lot about diagnostic testing procedures and analysis through AI. And in our next presentation by Thomas Grill from the company UX Focus, we will learn more about user experience in health and medtech. And we are already looking forward to your presentation, Thomas, and the webinar stage is already yours.
Hello, thank you. I hope everybody can hear me and I unmuted the microphone correctly. So my name is Thomas Drill and a warm welcome also from Linz in Austria. Yeah, UX Focus, what are we doing? As the name already says, we're focusing on the user and his experiences in a lot of different areas and also of course in medtech area. And I want you to show you a little bit about what the things that we are doing and give you also some examples. And maybe, as Patricia also already said, we are now not developing the new technology and we're not the experts in the algorithms and so itself, but we're focusing on the user who has really to use it. So what are our areas where we provide our services? It's basically 3 different areas. The first one is finding out what the users really need. So of course, there are a lot of perfect technologies that provide real correct answers and so on. But how can the user cope with that? How can he interact with different solutions? What information does the user need? So we're providing services in the area of user requirements analysis, where we're doing a lot of interviews, focus group, etc., and also participating in studies in order to support our customers in finding out what the user needs and what requirements the user really has. Based on these user requirements combined with the factual requirements, we have our second area where it's about typical user interface design, user experience design, service design, where we're supporting our customers in building user experience concepts, where our mission is always to have to focus on the user. So we are not just building concepts for products, but for products that the users really need and want, and developing, designing products in the course of a user-centered design approach. The third area is usability evaluations, where we can learn from the users. We can learn from people. As already mentioned in previous talks, there's a big need of studies, clinical studies, testing products, prototypes with users in order to find out if it works properly and if the designs that are basically shown to the users and the prototypes that are shown to the users are also perceived in the correct way. If you have a correct algorithm, provides the correct answers, it also needs to be shown to the user in the correct way. So what are we doing? User requirements analysis. It's important to know about the target groups. We're providing services in the area of target group definition, persona definition, defining who the user really is, whom to focus on, whom to use in order to prioritize their different requirements. We are supporting a goal-oriented product development. That means the goals are set by the users and basically help then the company in order to prioritize what to do and what to develop. This, of course, saves in cost in development because you're not developing something that nobody needs, and you're focusing on the real requirements of the users that can be tested and documented accordingly. Altogether, so our goal is to make the needs of the users, of the target groups, of your target groups actually really tangible and graspable, and we're developing different material based on interviews, focus group, etc., in order to develop the correct products. Second is then we're in the basically the development of UX concepts, user experience concepts, service design concepts. We're developing interactive products. We're developing the communication between a cool product, your cool product maybe, and the way how the user understands it. In the form basically of wireframes up to click prototypes, clickable prototypes which can be tested and already in a quite early stage. And then of course developed where we do working together with different development partners. Here, and the third one is the evaluation area. Why evaluating something? It's about getting insights in order to improve something, to improve a method, to improve user interface, to improve a tool. And if you already have a tool and you want to find out if it's good, that's a perfect way of finding it out, actually putting users onto the tool, having it used, and find out about usability issues, usage issues, and also issues regarding the content. So that's basically the three areas where we work in. So in order to show you some different projects and ideas where we are located in, I selected some few projects that also target a little bit the area of healthcare, medicine, medtech. One of the projects is where we work together with a company called DIGI-B, and it's located near Salzburg in Grödig, with the main headquarters in Switzerland. We developed this reader in the middle, which is a laboratory device where we focus on different analysis method and production and service software of these readers, where we also interviewed a lot different people from the users then in the end up to the ones producing it in order to find out about different workflows, in order to set up different measurement methods like absorbance analysis up to cell counting, over fluorescence imaging analysis methods that could be then configured and adjusted then with these devices correctly, which have then be tested and iterated accordingly. Another project, just swapping into the area of health actually, is the Move effect, which is basically a responsive application working on your computer desktop as well as on mobile phone. And the goal here is to use gamification methods in order to motivate people getting more active, more vital, and fast, also being more healthy, of course, which affects the whole style of living then. And yeah, we did a total redesign of all the application, put gamification methods points in there, and yeah, supported the company Move effect here with this product. Another project that I want to present you is before DIGI-B-CUBE, there was a prior support funding called Permidas, where we did 2 different other projects, small projects, where we were basically working in the area of user requirements analysis, where we did focus groups with, for example, Go who work basically together with laboratory staff and develop purification processes, and they have the prediction software Chromax, where we analyze the needs of the user, we develop personas for that, we develop needs and requirements for their software in order to lead it to the next level. Same here with our community, our community, where we did user requirements analysis, we did a focus group with different researchers, basically laboratory researchers, focusing on neoantigen recognition, where we figured out what the people in the laboratory really need, how the processes are, where they really stop over, and where needs appear from based on the software, but also based on the processes that they basically conduct in their daily activities. Here another project that's a research project, an FP7 project, focusing on the needs of developers actually developing for people with handicaps, where we did different studies. Here on the left side, we have basically a screenshot of a recording from a study where we tried out different interaction devices and figured out how people work in different scenarios. It was an FP7 project over 3 years actually, where we did a lot of prototyping, study design, user requirements definitions, prototyping, and yeah, also testing, of course. Yeah, so our services are basically focusing on the user. We always have the user in mind. We do workshops, we develop concepts, we're providing support in sketching, prototyping, different stages of developing basically a product up to clickable, testable prototyping, where we also do user tests and testing basically solutions that already exist or do not exist, depending on how far the development is. Okay, well, last slide. So basically my name is again Thomas Grill from UX Focus. We are located in Linz, and if there are any questions, feel free to contact me. Thank you.
Thank you, Thomas, also here for the presentation and for the input about what users really need and how you and your company could support in further collaboration projects in the framework of the DIGI-B-CUBE project. And I'm already using one of your tools. It's Move effect. And I can recommend to everybody of you who is living in Austria, go and test it. So thank you again. Our next speaker, Salvador Sales from the company CalSense SL, will give us a presentation on fiber optic platforms for medical application. So please, Salvador, we look forward to your presentation.
Okay, do you listen to me?
Yes, we hear you and we can see your screen. Maybe you can, make the presentation a little bit bigger. Thanks.
Yeah, no. So do you see now? Yeah. So good morning to everybody. Okay, let me go back. I cannot go. Sorry. I think I have some problems. It's okay?
Maybe you go back, or it's on one of the other screens where you have to click to the next slide.
Okay.
So yeah, you can do it also in this way. It's fine.
Well, if not, I will try to do it that way. Okay, yeah, fine.
Yeah,
perfect.
now it's working.
Okay, sorry. My company is a bit different to the other companies. And because well, I'm going to explain you a bit more. We are more or less expert on fiber optics for sensor, and let's see if we can find some applications also for medical application. So again, problems with what is happening. Okay, so this is our company. It was a spin-off company from the university, from the Universidad Politécnica de Valencia. And it was people coming from the Civil Engineering Research Institute and the Telecommunication Research Institute. And so we brought CalSense. And so from the Civil Engineer group, we bring the knowledge of materials, structural modeling, analysis, industrial applications. And from the Telecommunications group, we brought the experience from photonics, signal processing, or our artificial intelligence or communications. So the idea was for making a structural health monitoring mainly using fiber sensors, but of course we are using also other sensors if it is possible. So our main focus are civil and aerospace applications, and of course we have made something about medical, and maybe we can find some partners just to continue our way on these applications. So just to explain you a bit, just to show you our potentials. We have been working in European projects, just to mention some of the European projects that the people in CalSense are involved. So most of the projects are, this is a project related with communications, 5G communications. But the important thing is that we are using fibers, and special fibers that most of the people are not allowed to use it. But because we are working with big companies that make fibers, special fibers, so we can use it and then employ for sensors. Also, we have working in ERC, again, with new type of fibers. And in another project, just to make sensors for civil engineering applications, but also some of the applications are robotics or nuclear or pipelines. So this is our thing. Just to show you one of the things that we are doing is this. It's not exactly medical applications, but just to show you that we are installing sensors in all type of bridges, tunnels, or other things. And also we make, okay, this is our central control center. Now we are monitoring more than 500 sensors in 3 bridges online and synchronized. Just to, maybe I can try to show you a video. I don't know if you see. So this is just to show you that we are monitoring online how the train is coming and how the bridge is moving. Just to, let's say, to show a bit the potential that we can have. So this is what we can do it in civil engineering applications. So we can monitor and synchronize more than 500 sensors remotely, online, and all these things. Other things that we can do, that we have done in the past, it was just to show you our potential, is to work with sensors for special application. This was a special requirement for ESA, for the European Space Agency, that they needed a sensor that arrived till more than 1000 degrees, and quite linear and no hysteresis, quite repetitive. So we made a special sensor for them using FVGs or LPGs. I don't know if you know what is this, but it's a type of point sensor. But also we can work also with other type of sensors or fiber sensors, making other technologies like Rayleigh, bluing, or you know, or Raman scattering, or any type of technology. This is our facilities where we can also fabricate our own sensors, and so we can fabricate our own sensor for just for temperature, for strength, for vibration, for what people call 3D shape sensor, and using special fibers that maybe is too small and you cannot find in the market, but we know the providers, so we can contact them and they can make special fibers for us. Maybe this is important for mega applications, just to put this type of small fiber in the body of a person. So this goes for civil and space application, but also we have made something just for medical application that we can apply our technology. So just in a collaboration with the Medical Center of Groningen and the Twente University, we made for them these sensors in a fiber, in a special fiber, and then they put it in a catheter and endoscope just to help the surgeon to know the position, where is the catheter or the endoscope. So we can apply our sensor for this. So they have a requirement, they talk with us, so we can fabricate for them a special sensor. Also, we have made sensors for our application. I cannot show you because we have some NDAs, and I can show you the pictures, but there are applications for smart textiles, just to check even the health of the people, for dentist application, just to measure some inorganic substance, or to measure the pH or the refractive index variation, or just to measure the temperature in the body. Inside the body, we can, inside there, we put the fiber. And the idea finally is to have a fiber, and if we have some people that are experts on chemistry or in medical, we can put some thing in the fibers that you can help us with that, and then we can monitor if there is some virus or some molecules or whatever, because these molecules interact with the things that you put in the fiber, and we just checking the parameters of the fiber, and then of the light that passes through the fiber, and then we can diagnose if we have a virus or we have or whatever thing. So what we are doing is what we call lab on a fiber, and maybe you can also listen lab on a photonic chip. So I think both technologies are complementary, and it depends on at the end of the final application, because depending on different parameters like the cost or the accessibility to the point of where you can measure, or the number of sensors that you want to put it, it's better to use a photonic chip or use just an optical fiber. So we are working on optical fiber. Of course, we have some colleagues that work on photonic chip, and depending on the application, we can tell you, okay, this is better to do with this technology or without this technology. So this is what I just to tell you. We are not expert on medical or chemical applications, sorry, but I think we can help. We can make nice sensors in fiber optics. This was a picture that we can make different sensors at different points using special fibers. And if you want to know more about us, okay, just contact me, or just go to our webpage. In the webpage, there is applications of civil engineer or aerospace, but we have papers or we have other things that I can share with you. And that's it. I think it's enough. Okay. So you have any questions?
Perfect, Salvador. Thank you very much for the presentation. Also to see an application which is not normally used for the field of medical technology in this way. So as at the former speeches or presentations, please all questions into the chat. And I already have seen that there is one where somebody is asking about more information about the work with Groningen, if I'm pronouncing it well. So thank you again. And now I'm pleased to welcome our next speaker, after lectures on heart, lungs, and skin diagnostics, we now will come to our next organ, we can say. So Thomas Potrusil is currently in the process of funding his startup company, and he will now take us to a journey into the human brain. So Thomas, the webinar stage is yours. Thanks.
Thank you very much, Patricia. I also need to ask you if you can hear me and everything is okay, and if you can see my first slide.
Everything perfect.
Right, thank you very much. So my name is Thomas Potrusil. I'm located in Upper Austria, close to Linz. And as Patricia already said, I'm at the very, very beginning of my startup. And I want to use this platform today to introduce you and to give you an overview about our project. It's an application for early diagnosis of neurodegenerative diseases, and it's part of the last couple of years when I worked together with Professor Christoph Scherfel from the Medical University in Innsbruck. Early diagnosis, this brings me to the first slide. What does it mean? Actually, imagine you're sitting at home at a rainy Sunday together with your grandparents, drinking coffee, eating some cake, and then you suddenly realize some changes in the movement of your grandfather. For example, when he takes up his cup of coffee, you see some tremor, or you realized that there's some unusual slowness in the movement. So those are actually first hints of any neurodegenerative diseases. So all the alterations in example movement, they happen very slow. So consequently, these changes are not really recognized by the person concerned. And usually the family says, hey, come, let's go to the clinic and take a look to the specialist. Then they show up in the hospital at a movement disorder specialist. And what are they doing? Then, of course, they are first examining the patient history, since when some anomalies were detected by the family, and they made extensive clinical testing. They test the ability to move, so they made gait analysis. They test about the postural stability of the person, also the walking style, also if there's some kind of falling in the last couple of months. And with these tests, all the tests get some clinical scores, usually between 1 and 10, and these are then that leads to a first diagnosis of any disease. And of course, with this clinical scores, you test distinct areas within the brain. And that's the reason why also as a standard procedure, a magnetic resonance tomography brain scan is made. Of course, you can imagine all this procedure is quite time consuming. Many specialists are needed from different departments, neurologists, neuroradiologists. And since the scores are used in this testing environment, the results are always strongly observer dependent. And this leads me to the aim of this project. The aim was to develop a fully automated pipeline. It should be observer independent, and it should be standardized and reproducible across different platforms, MRT platforms. I mean, MRT platforms are dependent on their magnetic strengths. So there are 1.5 Tesla or 3.5 Tesla that are usually used in the clinics. And what we are doing is then this MRI analysis of the brain scan based on this first brain scan that is made when the possible patient is coming to the clinic. So usually at such a scan, there are several sequences scanned. So one is called a T1 weighted. And here on this picture, you see an axial view of this. So there are many two-dimensional images imaged, and the neuroradiologist can stack through it and gets also only by looking at those images to his results. What we are doing, we are using for this project, we used an open source software from Harvard Medical University, and it segments different parts of the brain. As you can here see with the red line, you get the pile surface, then you get separate the white matter. You can distinguish between the gray and the white matter. And after 8 hours of processing, you get out some subcortical and cortical volumes that are segmented. And this information, and you get out the three-dimensional surface model of the whole brain and the three-dimensional information, so the volume of every of those segmented subcortical and cortical brain regions. So here I will show you a slide about this that you can imagine it, what we are doing. So we have different, for this study, we analyze different subtypes of Parkinsonism diseased people and control. And you can easily see that the size of distinct brain regions are varying with different diseases. So for this study, we analyzed 22 subcortical volumes and used machine learning for pattern recognition and also for classification. And we got an early diagnostic accuracy for separating Parkinson diseased people from MSA or PSP of about 97%. Quite interesting is if you compare it to the clinical consensus criteria. So the experts in the clinical field only reach at this point, at this very beginning of the treatment, early diagnosis, they only reach about 63%. We are not only analyzing this structural MRT images, we also take a look into diffusion tensor imaging or about gradient echo. So diffusion tensor imaging, we can analyze the ADC or the FA, which are measures that represent the diffusion of the water molecules within the brain. So for example, if you have a specific region and you have a neurodegenerative process there, the missing, for example, gray matter is filled with fluid. And by analyzing those images, those sequences, we can get out a measure about the degree of the neurodegeneration. And the third picture, the R2 star, is based on a gradient echo. So we can also get a measure for the iron content within the brain. This is also of quite importance when diagnosing, for example, Alzheimer's disease. What we can do is then we take the segmented volumes and register them to the different sequences. And for the different volumes, segmented three-dimensional volumes, we get measures for the apparent diffusion coefficient, the fractional anisotropy, for example, or for the iron content. A big advantage, we obtain them quantitatively values of the individual diffusion space. So we can use those values for each from each patient to classify them. As an example, those are some regions that are highly significant different between those two groups. And in early diagnostic accuracy using machine learning, we get around an accuracy of 92%, but to separate those patients. Another thing that is based by diffusion tensor imaging is a global probabilistic tractography approach. So here we also use an open source software. It's called Dracula, also from Harvard. And you see here also pictures from a control, PSP and PSP patients. And you see some segmented white matter tracts with different colors here. These results are published in February, and we analyzed here patients, 6 white matter tracts and 3 subcortical volumes. And again, by using machine learning, learning the pattern, and also for classification, we got a separation accuracy of around 91% of those 3 groups. And please note that we have here a PSP P type, and the P type is a Parkinson type. That means at early diagnosis, it's quite hard to distinguish for the neurologist, for the specialist, between a PD patient and a PSP-P patient, because the symptoms are really, really similar. And after 2 years, they start to differ a little bit. So this is really a quite good result here. And another method we are using that could be interesting in the field of neurology, or is of interest, is we are also using the diffusion toolkit. This is also based on the diffusion tensor imaging sequence. And as you can see here, we get a nice picture about the connections within the brain. We can also feed in some 3-dimensionally segmented volumes and take a look about the connections that are established by those volumes and where they are going in the brain. Another thing is also, we can, for example, quantify the connection between 2 brain regions, and we can also visualize them. And this, I think, is also maybe of great interest for neurosurgeons that need to remove a tumor. We could make a full, or visualize a 3-dimensional model of patients' data and provide this for a first training, for example. Yes, my last slide, also matchmaking cooperation. What we need clinical institutions, clinical partners, also for collecting data, we can transform MR data to results, also providing results to you that can be published, for example. We are also interested in enrolling our presented prototype as an expert system to support your experts, meaning neurologists or radiologists, neuroradiologists. And we also need know-how in medical image processing, for example, 3-dimensional segmentation. The software, the open source software I presented to you is established in the scientific community. It's working quite well, but it has some disadvantages. And therefore, we are actually working now on a prototype by using deep learning image processing to build our own system. And for this, we also need we have medical image processing know-how, but it would be of interest for us to have more expertise in this field. And also, what can we offer for you as a partner? We have a strong focus in machine learning, AI, all the different subfields. So in pattern recognition, anomaly detection, classification, not only medical field, also in industry, time series analysis, also working on some university projects with deep reinforcement learning with the Institute for Machine Learning of Sepp Hochreiter, that he's located here at the JKU in Linz. And of course, we also do a lot of signal analysis. We can offer image processing or knowledge in medical image processing, and also in general in mathematical modeling or simulation of biological processes. I would be really happy if I can collaborate with you. And yes, thank you very much for your attention.
Thank you, Thomas, for the great presentation. And now we know a little bit more about neurodegenerative diseases. It's not that easy.
Okay.
Okay.
Yeah. If you have any
So,
questions, don't hesitate contacting me. I'm looking
okay,
forward,
perfect.
really. Okay.
Perfect. I was taking some notes. It was very interesting, and maybe, yeah, as before, all question about the diagnostic method of Thomas, please questions into the chat, and also if somebody wants to collaborate with him, also, yeah, write us and also the questions and the inputs into the chat. So now may I ask our next speaker for his presentation? It's Michael Meyer-Huber-Reinhardshuber of the company KML Vision, and he will now tell us more about the platform ECOSA. So please, dear Michael or Michael, the webinar stage is up to you. Thanks.
Yeah, thank you very much, Patricia. I hope everything is working fine and you could see my slide and hear me.
Everything perfect. Go on.
Great. Thank you very much. So my name is Michael Meyerhofer. I'm one of the co-founders and managing director of the Graz-based company KML Vision. And today I want to tell you a little bit about our core product, which is ECOSA, a flexible automated image analysis platform for decision support. So to get the background, the passion of our company lies in image data management and automated image analysis, which we provide for the two fields, life sciences and for healthcare. And as you all know that in these fields, visual inspections play a crucial role, for example, in R&D processes for decision support, also for quality control and assurance. And the problems there can be rather diverse. So usually you have different samples and you want to know, for example, the number of objects within a sample. You want to know in which region specific objects exist, for example, cells or pathologically altered tissue, or you want to classify different objects. And as you all know, a lot of things are still done manually by taking a look at the samples under microscope or so on, which have, of course, different problems. It can be slow, of course, error-prone, subjective, and rather expensive. But we also know that a lot of the things are digitized already, and there are often already digital images present of these samples. But with the digital images, of course, other questions arise. For example, how to manage all the data you are dealing with, how to organize the images, how to create, how to store them, how to create backups, and also how to trace who is doing what and when with the images, which is of special importance in the field of in fields related to medicine or life sciences. So there are also problems when going digital, especially when thinking of the availability, scalability, usability, and especially the flexibility of such a system. And for us, it was clear that we want to develop a solution for exactly these problems. And this solution we are developing is called ECOSA. It's a web-based platform, a software platform for these two things, image data management and automated image analysis, which is actually based on two core technologies, that is cloud computing and the usage of artificial intelligence, machine learning. Here on the left-hand side, you have an overview of the usual workflow when dealing or when using the platform ECOSA. So in the first two hexagons, you see samples are prepared and images are recorded. This can happen in different ways. For example, microscopy slides, digital cameras or microscopes, or really whole slide scanners. But you can also use like X-ray images or fluorescent images whatsoever you're taking your images with. Then ECOSA comes into play into this with these bright green hexagons. You can store your images on the platform. You can do, of course, the management, the viewing. you can annotate and highlight interesting objects and work together in this system in a multi-user environment. So it's has the advantage that you can use all these multi-user functionalities to work together on the platform. We support arbitrary image sizes and formats, so you can use standard images, but also, for example, whole slide images that are really, really big. For this, we provide also a tile-based image viewer, which works kind of like Google Maps. We offer convenient annotation tools in the system. And then we come to these darker green hexagons, which is more or less the core of our platform. There we provide specifically trained artificial intelligence applications to fulfill analysis tasks like counting specific cell types or finding specific regions in images. Also, it is really important to mention this part completely down on the left-hand slide, this integration of third-party software and individual module development. So we provide an open API to our system so that it is easy to connect to our platform, and we also do tailored module development for specific use cases. So if we take a closer look to this light green part, which is actually all this image data management, this we call ECOSA Portal, which is this image data management system that provides a multi-user graphical user interface. It's independent of the operating system since it can be accessed via the browser, the web browser. You can use annotations and labels to get the most out of your image data. So here you can see how the layout of the graphical user interface looks like when, for example, viewing a whole slide image and H&E stained tissue section, for example. As I said, we provide this fast tile-based viewer, and we also support whole slide images there. Then, of course, you can use these annotation tools to highlight specific regions. There exists, of course, the opportunity to show what other users, for example, assign labels for specific annotations. This is rather important to create a highly curated data set that we use afterwards for training an AI-based algorithm. You can also review all the annotations and labels that you set within the system, so you get the gallery view of all the annotations within a project. You can make a fast review of who assigned which label to which annotations. All this is perfectly set up to create a high curated data set with high quality for the following AI training. Then this darker green part is the part where all these AI-based algorithms come into play. This we call ECOSA Prisma. It is the part of automated analysis. With the applications, we provide fast and reproducible results. It's a module-based system, so it's easy to integrate new applications. And of course, the modules can be also tailored for the needs of our customers. The generic analysis workflow looks like this. So usually images are submitted to the system. Then, of course, the progress of the analysis can be monitored. As soon as the analysis results are ready, the results can be collected and insights into the system can be generated. Insights into actually the research questions you are looking at. Well, if we go further on, this whole application or this whole platform for providing the image analysis application is developed in a way that it is really, really easy to plug in new analysis modules into the system. This can be either done by us, which we provide through our online system, but it can be also done by our customers if they deploy the system at their own premises. So you see, with having an AI-based algorithm and adding some frame in the form of a Docker container, you get the Prisma app that can be plugged into the Prisma core and is available right away on the scalable platform. This looks like this in the graphical user phase. Then you can access the fully automated image analysis application in a really convenient way. You can monitor the progress, and then you can download, of course, the individual or merged results. On the left-hand side of the screenshot, you can see the analysis results, for example, of an image showing transmission electron microscopy. So an image taken with transmission electron microscopy from myocardial tissue, where a quantitative analysis of all the structures was performed. Well, as an example how fast this can be done to integrate new applications into our system, in the last weeks we integrated, for example, an application that is capable of detecting alterations in lung chest x-rays that happen due to COVID-19 detection. This algorithm, for example, we did not develop by ourselves. It was developed by some Canadian research teams. They provided it open source. However, it was a GitLab repository, and not everybody is capable, of course, of setting up all the systems required to use this source code in a way that you can apply it to your images. So we plugged it in as a module into our COSA system, and now it's possible for everybody just with a few clicks in a graphical user interface to apply this algorithm to images and to support research in this context. Well, the interface to our analysis backend we provide as an open API standard. So it's really easy to connect your devices or your clients or your apps to our analysis backend. So in this way, we open the opportunity to a really easy collaboration with our company and with our product. Because, for example, if you think of developing your own, let's say, for example, smartphone application, it's easy to send an image to our backend where we provide a scalable analysis algorithm in order for you to get back the result as soon as fast as possible. Well, we developed a diverse set of applications already, so from in vitro angiogenesis assays to, for example, applications in digital pathology, also some applications regarding fluorescence microscopy, retinal fundus imaging, and so on. So we are not restricted to special imaging modalities, for example. We can plug in any analysis algorithm you can think of into the system. Well, we're currently active in the DACH region, but have several collaboration partners also throughout Europe, and also some international customers from outside Europe already. And yeah, maybe we can find other collaboration partners also through this DIGI-B-CUBE project. Would be glad to hear something from you. Thank you very much for this opportunity. And if you have any questions, please write them in the chat or get in touch directly with us via email. And of course, you can also try out our platform for free with this link on the right-hand side. Thank you very much.
Thank you, Michael, for your presentation. And as already mentioned, all questions will go to the chat. So now I want to share my screen again with you. So we have now also reached the end of our webinar today. So thanks to all speakers and thanks to all of you for participating in this event. And we would be also happy to welcome you at our events in autumn in real life. And if you have any questions regarding the project, submission, or the call, we are available at info@digibcube.au. And otherwise, you have also the email addresses of me and of Gavel. Don't hesitate to contact us. And thanks again for your participation, and stay safe. We look forward for your proposals, of course. Thanks and bye. Have a nice week. Bye.
Bye. See
Bye.
you.
Goodbye.
Thanks.